Hi, I'm Marin Brewer. I'm a professor at the University of Georgia, and I'm in the Department of Plant Pathology. So, thank you for coming to my presentation. Traditionally, my research focuses on emerging plant diseases that are caused by fungi. But recently, I've been working on plant associated fungi that cause human diseases. And these ones are particularly problematic because they are developing resistance to fungicides in agricultural environments where they are found. So, today I'm going to be telling you a bit about how fungicides affect a particular fungus, Aspergillus fumigatus, that's found in the environment. And this fungus is developing resistance to these fungicides. And this is a big problem for human health. And I'll tell you a lot about that as I go through my presentation. So, there are several questions that I hope to answer during this presentation. The first being, why are Aspergillus fumigatus? And Aspergillus fumigatus is actually this fungus that's shown in the background here. This is a microscopic slide of it. But why is it a problem? And why is the disease it causes, aspergillosis, a concern for public health? And then what evidence do we have that this resistance, this antifungal resistance, is developing in agricultural environments? And then where are the hot spots for antifungal resistance? What types of agricultural environments are we seeing a lot of this developing in? And then what can we do as scientists, policymakers, crop advisors, and growers to better understand what's going on and also mitigate this issue of antifungal resistance developing in agricultural environments? So, Aspergillus fumigatus is a plant associated human pathogen. It's not a plant pathogen. It lives in the environment and it's normally found associated with plant debris. It's really good at decomposing organic matter. So, that's why we find a lot of it associated with soil and crop debris. And it's thermotolerant, which makes it survive very well in the human host. So it's a really bad pathogen of people because not only can it survive in really high temperatures in the environment, such as compost, but when it gets into people's bodies, if someone's immunocompromised, it can survive in that higher body temperature as well. So it's abundant in compost and it's acquired from the environment. So when people get infected with this, they breathe in spores from the environment. It's not contagious, it's not transmitted from person to person. It always is acquired from the environment. And people who are immunocompromised or have lung disease are the most susceptible. So, as this figure kind of shows, this fungus is everywhere in the environment. Most of us are breathing in, it's estimated probably about 100 spores of it per day, but people who are immunocompromised are the ones who are likely to get infected. So, aspergillosis is a, a spectrum of respiratory diseases caused by different Aspergillus species. And Aspergillus fumigatus is the most common fungus that causes aspergillosis. And again, it's acquired by inhaling spores of this fungus. And what this shows is the different types of diseases caused by Aspergillus fumigatus. So, healthy patients can have more mild conditions, such as asthma, and then people with chronic lung disease can develop um, chronic chronic pulmonary aspergillosis. And then people who are severely immunocompromised can develop invasive aspergillosis. So, this arrow kind of shows the direction of more severe disease, but also the direction of the condition being more rare. So, aspergillosis affects humans, it also affects other animals, and it's treated using azole drugs. So, really, the biggest problem with aspergillosis is the invasive form, which infects people who are immunocompromised from having organ transplants, also cancer patients that are undergoing chemotherapy, and then people with severe lung disease, such as cystic fibrosis. It's also becoming more of a problem in people with COVID-19. So, again, azole drugs are the best form of treatment for aspergillosis. But in both the environment and in patients, there's this problem of azole resistance developing. So azoles are also demethylation inhibitors, is what we call the fungicide form. These are used to combat plant pathogenic fungi in the environment. And here you can see the application of fungicides in the environment. And then here, this is an Aspergillus fumigatus growing in lung tissue. So the same type of drug, these azoles, are used in both environments. And they're really good at inhibiting fungi because they target this enzyme, CYP51A, that helps the fungus make ergosterol, which is really important for the fungal cell. So when azoles are used, the fungus is killed and it can't grow. But resistance has been developing both in patients and the environment. And now the fungus is able to survive even in the presence of these azoles. So, the way that this works is that the fungus develops mutations in that enzyme that's targeted by azoles. So, that enzyme is CYP51A. And in patients that are treated for long periods of time with azoles, that usually what happens is they develop a mutation, and then the mutated form of the enzyme is no longer susceptible to the azole. So the azole doesn't bind properly and the fungus becomes resistant. It no longer is affected by the azole drugs. But now we're finding some patients acquire Aspergillus fumigatus and they're already azole resistant and they haven't even been treated with azole drugs. So, what we're finding in these patients is they have a different type of mutation. And I'm just going to show you this image of kind of what that gene might look like. So, this just shows what that CYP51A, what the enzyme looks like, or the gene that encodes the enzyme. And this is what a sensitive one would look like. It's just an example of it. But what we're seeing in these isolates of Aspergillus fumigatus in patients that are acquiring resistant infections is they have what's called a tandem repeat. And that's what that TR stands for. So some will have a tandem repeat of 46 different DNA sequences, and then other ones have one of 34. And all this does is it allows the fungus to make more and more of that enzyme. So in this situation, not only does the azole bind very well to the enzyme, the fungus is making so much of it that the azoles aren't effective and the fungus is resistant. And the isolates that have these TR mutations end up being what's called pan-azole resistant, meaning they're resistant to many, many different types of azoles. So azoles that are used in hospitals to treat patients, azoles that are used in the environment, such as tebuconazole, as fungicides. And that's what we call this. We call it panazole resistance, meaning they're cross resistant. So this is what tebuconazole looks like. And the main thing I'm trying to show here is that the fungicide tebuconazole and the antifungal used to treat people infected with fungi, voriconazole, have very similar structures here. And this is why this cross resistance happens is once a fungus is resistant to tebuconazole, they're also resistant to the medical drug voriconazole. And they have these same sorts of structures. Sorry, it's actually this one over here that I meant to be pointing to. So they have this, um, this is why it has the term triazole. So both voriconazole and tebuconazole have the same effect on the fungus. And if it develops resistance to one, it's resistant to the other. And that's this cross resistance. But what's really scary is these patients that have invasive aspergillosis. If their infection, if the fungus is azole resistant, the mortality is very high. It's almost 100%. So people who have invasive aspergillosis and it's resistant will most likely die if if they can't be treated, which they usually can't with these resistant infections. So this diagram here shows what happens when people acquire a susceptible infection and then develop resistance. So the fungus lives out in the environment on plant material and it's susceptible to azoles. So when the person first acquires the infection, they're treated with azoles, but over time the fungus will perhaps mutate and then become resistant. So here now the fungus is resistant. But what happens when the fungus develops resistance in the environment is that it's exposed to these azole fungicides in the environment and it develops resistant in the environment. So when the immunocompromised individual inhales those spores, the fungus is already resistant. And this is when it's really problematic and the person can't be treated. And what they're finding is there's a lot of evidence that this is happening where resistance isn't just developing in patients treated with azoles, but it's developing actually in the environment due to the use of azole to treat plant pathogenic fungi. So the first line of evidence is that these patients who've never been exposed to azoles have these panazole resistant infections. So this indicates that they acquired a resistant fungus from the environment. And they're finding that these fungi in the environment and in patients have the same types of mutations. These ones that are resistant and they're resistant to many different types of azoles all have these tandem repeat or TR mutations. And they're also finding that isolates in people and isolates from the environment that are resistant have the same multi-locus genotype. So this just means they have like the same DNA fingerprint. And this shows that they're related. So there's the same strains or isolates in the environment that are in people. And this is just more evidence that they're coming from the environment. But some people aren't convinced that azole resistant Aspergillus fumigatus, this is what ARF means, which direction it's coming. So, did it develop in people and then it got into the environment, or is it developing in the environment and moving into people? So, this is something we were interested in looking at. So we first started off looking at isolates, collecting them from agricultural environments in the Southeast. And we started with Georgia and Florida. And this work was supported by funds from the Centers for Disease Control and Prevention, the CDC. So we went out and collected about 700 different isolates from 50 different agricultural sites where azoles are used for crop protection. And we collected soil and plant debris from fields, orchards, where peanuts were grown, grapes, pecan, apples, strawberries, and other crops. And we then did a second set of sampling the following couple of years, looking at the East Coast and the West Coast of the US to include additional samples. And now additional studies are actually being done in the central part of the US, so nobody's being left out. But the sites here, we sampled 52 different sites and expanded to different crops, including tulip, hemp, wheat, apple, and the other crops listed here. And then we also sampled an organic farm and compost, organic compost from Georgia to compare with these environments where azoles are used. And in this set of sampling, we had 727 isolates. So, of all the isolates we collected, about half or 350, actually, it's less than half, but 350, we screened them for resistance to tebuconazole, the fungicide, and then to other azole antifungal drugs. And what we found is that in the first study with the isolates from Georgia and Florida, we had 12 that were resistant, and they came from a compost pile and from pecan debris. And these panazole resistant isolates, they were resistant to tebuconazole and multiple medical azoles had that tandem repeat that was 46 DNA bases long. And then in the East and West Coast study, we ended up with 20 different azole resistant isolates that had both the TR46 and TR34 mutations for resistance. And these came from sites where they were growing grape, wheat, herbs, peach, tulips, and also compost again. So we wanted to then see with these isolates now that we had, can we determine if they're moving from agricultural environments into humans? And I was actually reading this book at the time called Big Chicken, and this book is by Marin McKenna, and it's a similar story when we started identifying antibiotic resistance in bacteria that were causing disease in people. And people thought that, well, maybe it's moving from farm animals from chicken into people because we're seeing a lot of these resistant bacteria also in chickens. And there was the same problem. They couldn't determine whether or not the resistant bacteria were moving from chickens into humans or from humans into chickens. And the way they were able to kind of pinpoint it in the end is they found resistance in the bacteria that were in people, but it was resistance to an antibiotic only used in chicken. So that kind of showed since it's never used in people, they didn't develop it. It was from the chickens and moved into people. So this gave me the idea to look for a signature that would show that these resistant isolates collected from humans are coming from agriculture. So, do these azole resistant isolates in people have resistance to other fungicide classes? So the fungicide that I thought would be really good to look at would be the QOIs or the strobilurins. So these aren't used to treat people, but they're used in ag environments. So if we look for resistance to QOIs in Aspergillus fumigatus, found in people, this would indicate that they've been exposed in agricultural environments. So there's this mutation that makes strains of Aspergillus or makes strains of all fungi actually resistance to QOIs, and it's called G143A, and it's in this particular gene. And if a fungus has this G143A mutation in the gene cytochrome B, they're resistant to QOIs. And then there's other particular markers we were looking at too, but we were able to look through the genomes of these isolates. So we have a lot of genome sequence, and we look through environmental isolates, human isolates. All of this is available publicly on large databases. And we looked for resistance to other classes of fungicide, including the methylbenzimidazole carbamates, and then also SDHIs, the succinate dehydrogenase inhibitor. So these are all different classes of fungicides. And what we found when we looked at the isolates, and they're shown here, is that many different isolates that were sensitive. So what we're showing here is this is CYP51 for azole resistance. They were wild type, which meant they were sensitive. And then for QOI, sensitive, methylbenzimidazole carbamate, sensitive, SDHI, sensitive. So they were all sensitive if they were not azole resistant. But when we looked at all the different isolates at this time that we could scan from the environment and from humans, from India, the Netherlands, and the USA, we found that in the isolates that had the TR34 or TR46, making them resistant to azoles, a lot of them showed that mutation that makes them resistant to QOIs. Many of them had the mutation that makes them resistant to methyl benzimidazole carbamates. And then a couple had resistance to the SDHIs. So, what this tells us is these pan azole resistant Aspergillus fumigatus are multi-fungicide resistant. So not only are they resistant to azoles, they're resistant to all these other fungicides. And these aren't just isolates in the environment, these are isolates in humans. So this was really interesting because it shows that multi-fungicide resistant Aspergillus fumigatus is in the environment and humans, and that this was geographically widespread. So these agricultural fungicide resistance markers, so this G143A and the H270Y for both of these fungicides that aren't used in people, but finding them in isolates from people supports that these isolates are acquiring azole resistance in the environment and then moving into people. So we were able to show that it does seem to be coming from agricultural environments. And at this point, we were interested in finding out which environments are the hot spots for resistance. So, a student in my lab, Caroline Burks, was a master's student at the time, and she went through all published reports of azole resistant Aspergillus fumigatus. And she only looked at environmental samples, not ones from patients or other animals. So all of these environmental samples are assumed to be saprophytic, so living on organic matter or decaying plants. And she ended up with a total of 1,292 of these isolates from the environment from 52 different papers that had been published. And so when she looked at which regions these came from, you can see here the vast majority of resistance is found in Europe. And that's where most of these studies have actually been done. So there is a bit of sampling bias here, but it does seem like most of the high levels of resistance they're seeing is in Europe. And right now, in North America, there haven't been a lot of studies done. And at this point, we aren't seeing as much resistance in people that have developed aspergillosis as they have in Europe. So now is a good time to start looking into how we can mitigate this issue before it becomes a bigger problem here. And then Caroline also looked at what type of environment they were found in. Was it developed environments, which include things like hospitals, um, places where people live, gardens, and then what she found in the developed environments where most came from. So about 47.6% of the resistant isolates came from developed environments. And those were mostly from hospitals, so from the air in a hospital or from a flower garden. But a large amount, about 35%, came from agricultural environments. And interestingly, most of these were from flower farms. And then when she looked at the different substrates that they came from, most of them came from soil. So a lot of the sampling comes from soil and then from air and then plant debris. But it's really important to look at air because that's where people are acquiring this. They're getting it from the air by breathing it in. So additional sampling of air is something we're really interested in doing. But based on the analyses Caroline did, and then other studies that we've looked at, and ongoing work we've done in our lab, looking at, we've looked at compost, soil, and tulip bulbs. We've found that these retail products, not necessarily crops or food from produce, but mostly lawn products have a lot of resistant Aspergillus fumigatus. So these hot spots seem to be flower gardens and flower farms. Compost is a place where they're finding a lot of resistance. And then retail compost and retail flower bulbs. So these seem to be the big hot spots. And we're interested in finding out more, what is it about these environments that make them hotspots for azole-resistant Aspergillus fumigatus? But we have also found azole-resistant Aspergillus fumigatus in vineyards, wheat fields, peach and apple orchards, and in raw peanuts. So these are also areas, although they don't have as much as what we're finding in the hotspots, they're areas of concern. So one thing that's interesting, and I had mentioned how they find a lot more azole-resistant fumigatus in Europe, is that that's where most of the azoles in agriculture are being used. We don't use as much in North America, so that's promising. But a study done by the CDC shows that agricultural use in the US has gone up over fourfold since 2006. So it's increasing drastically. So there's a lot more azoles that are being used. So this is pretty concerning. We don't want to increase azole resistant Aspergillus fumigatus. So using azoles and other fungicides for things like optimizing plant health or ensuring that your plants don't get any sorts of disease should be avoided. Using azoles when there's a serious threat of disease or your plants are looking like they are going to get infected, that's a different story. But we definitely need to be stewards of these azoles to mitigate this problem. Although azoles are very important for food security, there's huge reductions in yield from plants from fungi. They're the number one disease causing organism. We need these compounds to produce food, especially they're important for food safety as well with mycotoxins. So we need them for mycotoxin reduction, They're very effective, they're very inexpensive, and they're broad spectrum against a lot of different plant pathogenic fungi. And one thing actually with the azoles that's really important compared to some of these other fungicides I've mentioned, such as the methylbenzimidazole carbamates, the QOIs, and the SDHIs, is that resistance doesn't actually develop as rapidly to azoles as these other ones. So azoles are really important for plant protection, and we need to continue to be able to use them. But there's things we can do so we can preserve them to use in agriculture and to use them for as antifungals for infections that are caused by fungi in people. So, one of the things that's really important that we try to do the best we can is use integrated pest management to reduce fungicide inputs. So, planting disease resistant varieties, monitoring for disease and also for resistance, and using fungicides judiciously. If there's resistance to one particular group, then use a different class. Use alternative control strategies such as biocontrol, and then minimize the fungicide applications to only using them when they're needed. And one of the more promising things I've seen a lot of lately is using precision spray technology where only the plant where there's a lot of exposed plant tissue is sprayed, and the amount of fungicide in the environment is minimized. And then, of course, another really important thing to do is to educate farmers, industry specialists, extension agents, commodity groups, and other scientists about this threat. A lot of people aren't aware of this problem. So I've been spending a lot of time lately actually doing things like this, talking with people, letting people know that this is a problem and that fungicides don't only just lead to resistance in plant pathogenic fungi, but they're leading to resistance in these non target species, such as fungi associated with plants that cause human diseases. And there are other fungi that we're concerned about, not just Aspergillus fumigatus, that can be associated with plants. So we're looking into those as well to make sure they're not developing resistance. Because in human health, there are only three main classes of antifungi or antifungals that can be used. So it's really important to preserve these azoles because they work so well for so many fungal infections. So by increasing awareness, um, I've worked with Dr. Kirsten Wise to write some articles to get out there and just communicate with people. So not just scientists are aware, but extension agents and growers are learning about this issue. And then there's a podcast that I recently had. And then I also communicated with a lot of scientists at the plant pathology meeting, Plant Health in 2021. I gave the plenary talk to communicate about this. So, other things that we can do to minimize the risk of antifungal resistance development is to continue surveying and monitoring the environment to see where we're seeing hotspots and try to figure out how to minimize the development of resistance in these hotspots. Also act as stewards of the environment. I mentioned the use of using single site fungicides prudently for disease control, and then improving and recommending integrated pest management strategies. One of the more interesting things is to look at these hot spots like debris piles, waste piles, and compost and figure out the best way to manage them so additional resistant spores aren't spreading into the environment. And then, of course, continuing to spread awareness, like I just mentioned, by communicating with people and educating people about this issue. And really, this issue of antifungal resistance needs a One Health approach where we consider the effects on human health, environmental health, and animal health, and recognize this interconnection between people, animals, plants, and the environment. And we have to use a collaborative approach where we're working at the local, regional, national, and global levels together with people who are involved in agriculture, with scientists, with doctors, veterinarians, all working together so that we can preserve these important compounds. And here I'm focusing specifically on azoles, but preserving azoles and fungicides and antifungals so we can continue to use them to protect people and to protect plants. So thank you.